Hello, good afternoon to everyone. My name is Noemi Parraga-Nino. I'm a postdoctoral researcher at the German Trias Pujol Research Institute. I'm member of the Clinical and Environmental Infectious Disease Study Group, and I'm responsible of Legionella Disease Study Group of the same research center. Thank you so much for the organizers for trusting me to give this speech that is entitled Persistence of Viable but Non-Culturable Legionella Pneumophila State in Hospital Water System, a Hidden Enemy. This work has been published in Science of Total Environment just 2 months ago. First of all, I want to introduce you a little bit about Legionella. Legionella is a gram-negative bacteria that naturally lives in some wet sites such as rivers, ponds, and lakes, but we can find these bacteria as well in wet soil, as for example Legionella Long Beach. And these environmental places, we can find Legionella in a very low concentrations. And the problem is when these bacteria, and there's no risk of this kind of concentration. The problem is when these bacteria enters to man-made water systems, such as fountains, water facilities, or cooling towers, swimming pools. In these places, Legionella find the temperature that it needs to grow, to exponentially growth, achieving very high concentrations. The danger for human being is not only being in contact with this installation or facility that it's colonized, but as well if this facility produce aerosols. The infection of human being is by the inhalation of these aerosols containing Legionella. These bacteria that by the inhalation arise to the low respiratory tract, and is when the infection of the alveolar macrophages it's produced. Here you can see us in the infection. Legionella has a parasitic life, and it grows exponentially inside this macrophage. In the other part, in the environmental life of bacteria, we can find the bacteria in three different states. First one is the free-living Legionella that is in a planktonic state. We can find as well Legionella infecting amoeba, that it has the same behavior than in the infection of the macrophage, but we can find as well Legionella being part of biofilms. In these two states, in these last two states, like in amoeba or in the biofilm, Legionella it has an extra protection against some stressors such as biocides or temperatures. The infection of Legionella, it occurs only through contact with the colonized environmental focus. It hasn't been described any contagion person to person. If we are infected with Legionella, we can suffer two different kind of illness. First of all is the pontic fever. That is characterized such as like a flu symptoms such as muscle pain, chills, or headache. And in this case, the incubation time is between 24 or 48 hours. In the other case, normally, if the person has some comorbidities, some it's a person at risk, it's an old person, it has some other illness, if the person smokes or this kind of factors, risk factors, this person can develop Legionella disease that is characterized with pneumonia or other extrapulmonary symptoms as endocarditis. In this case, the incubation time is a little bit longer. It's between 2 and 10 days post-exposure, and it requires special antibiotics because of the intracellular state of Legionella. As all cases of Legionella disease has to be reported to health authorities, we have a lot of information about the incidence of this illness. Since in the last 3 decades, the incidence of Legionella has increased a lot. It has multiplied by 5 the incidence. we can see here a first increase in 2000 that we think that it's because of the appearance of new diagnostic tools such as antigenuria, and that it did very easy to detect a patient with Legionella disease only by using urine samples. Later, we can see here another increase in 2016, that we think that in this case, this increase is a little bit multifactorial. It's because of the clinicians are more aware about this illness. We think as well that there are more environmental sources colonized by this bacteria, and as well because we have more population at risk. The incidence, the last incidence described, it's 2.5, but this incidence is not homogeneous in all countries in Europe. For example, in Spain, we have a high incidence, higher than 3 point, it's between 4 and 5, while in other countries they have a really, really low incidence. It's for that reason, the high incidence that we had in Spain, that we have a legislation about all the activities that we have to perform that are mandatory in order to control and prevent Legionnaires or Legionella colonization in our facilities. In these legislations, it's regulated all the analytics that we have to perform, the temperatures that we have to achieve in different points of our installations, and all the disinfections that we have to perform as well in our facilities. To understand all these requirements, I'm going to explain a little bit about the temperature and the relation with Legionella. The optimal growth of Legionella is between 35 and 37 degrees, but it can grow between 20 and 45 degrees. Below 20 degrees, Legionella is not going to replicate, but it's going to be alive. And from 45 to almost 70 degrees, it as well is not going to replicate, but it's going to be alive. But above 70 degrees, Legionella is going to die. So I've explained these temperatures because it's very, very related with our work that I've said before, persistence of viable but non-culturable Legionella pneumophila state in hospital water systems, hidden enemy. So, material of methods of this work. We have performed this study in a hospital in Barcelona, a very big hospital with two independent hot water installations. One of these hot water installations is that we have called high zone, that is this part in red. And the other one is the low zone, that is in gray. And as you can see, it's much higher, much longer and much bigger than the one in red. And you're going to understand a little bit later why I'm commenting this aspect. This study was performed through 6 years. It was a longitudinal study from 2012 to 2017. And we studied 80 different isolates by genotyping, by post-Pujol electrophoresis, and 9 of these isolates were later studied by thermotolerance. Now I'm going to explain a little bit this aspect. During these 6 years, we analyzed 50 positive water samples. There were plenty of other samples, but they were negative, so we used these ones that were positive. And from here, we obtained 80 different isolates. These isolates were studied by post-Pujol electrophoresis, that is that technique where we study different patterns of the genome when we use restriction enzymes to code it, no? And we obtain different patterns. By studying these isolates by post-Pujol electrophoresis, we obtained that all these isolates had 3 different patterns. So we can classify these isolates in 3 different patterns. And we choose 9 of these 80 isolates to do the thermal tolerance study. So we try to take isolates from each pattern to obtain to study if there were difference between patterns or not. First of all, this is the result that we obtained by the study of buffet electrophoresis. We obtained that high zones installations were predominantly colonized by pattern V, while in low zones we have predominantly pattern A, as you can see here. And this distribution was maintained through the years of the study. As I mentioned before, high installation, the high zone installation was shorter, and this is why we could obtain medium temperatures higher than in the low installation. In high zone installation, we obtain a medium temperature in the hot water system, about 56.8 degrees, while in the low zone installation, we obtain 54.5 temperature, the medium temperature. Okay, later what we performed was the thermic treatments and the study of the thermotolerance. What we did here is to choose 9 different isolates from the 3 different patterns, and we performed 24 hours heat treatments at 3 different temperatures, 50, 55, and 60 degrees. What we did is to do culture plates of the pre-treatment, post-treatment, and check what we obtained. What we obtained was this. After the treatment, we had a known concentration, but after the treatment, we couldn't obtain any color in culture plate. So, by treatments at 50, 55, and 60 degrees, 24 hours, we could eliminate all culturable bacteria. What we wanted to study later was the viability of these bacteria after the treatment. So we decided to study by flow cytometry. When we perform a flow cytometry in bacteria, it's difficult because bacteria are very small. It's difficult to analyze only by shape, by surface, by size. So it's difficult. So we have to use some dyes. In this study, we use statein. That statein is used to dye total bacteria. It's permeable to the membrane, so it's going to dye all bacteria. It's our control marker of bacteria. But later we use as well in combination live or dye. That is a dye that is going to enter only in the bacteria if the membrane is damaged. So it's the way to differentiate. For example, bacteria that are going to appear in this section of our window, no, in the flow cytometry, they're going to be cytopositive, that it means that it's bacteria, and lot positive. It's going to this is going to mean that this bacteria has the membrane damage, so it's going to have the membrane integrity damage, so it's not viable. But the ones that are going to be here that are statein positive, they are bacteria because they are dye, well, they're with the dye statein, but they don't have the live or dye, sorry, because it's a little bit difficult to understand. This means that they are viable because this dye hasn't gone inside the cell. So we obtain after the treatment the same temperatures 50, 55, and 50 and 60, sorry. During 24 hours, we obtained these results. At 60 degrees, we couldn't obtain any recover of bacteria. So I think that the 10%, the percentage that we have here is on maybe the detection limit. But if we observe this graph, what we see is that after treatment at 50 degrees, we obtain between 50 and 60% of the bacteria that were still alive. And by increasing the temperature to 55 degrees, we obtain between 20 and 30% of recover, or the bacteria were still alive. So After this treatment, although we didn't obtain any colon, we observed that at least at 50, there were more 50% of the bacteria were still alive. So by studying our installation only by culture plate, that it's a technique that it's mandatory by the legislation to control the installations, we are not detecting this subpopulation. So the conclusions of our work is that the same strain of Legionella pneumophila persists in hospital hot water system for years. Keeping hot water system temperature at 50 degrees for 24 hours in distal point will allow the survival of replicative Legionella pneumophila, although culture bacteria would not be detected. The increase of this temperature 5 degrees in distal points could reduce the presence of Legionella by more than 25%. Viable but non-culturable Legionella pneumophila are detected at temperatures that are required by law in installations, and Legionella surveillance through the standard culture underestimates its presence. So I want to thank my workmates in the Legionnaire's disease study group, and as well to all my workmates at the clinical and environmental infectious disease study group. Thank you so much for your attention, and I'm going to be here to attend any question or comment that you have of my presentation. Thank you so much.